All right, so we finished talking about meiosis. Now in this video, we're going to compare the process of meiosis and mitosis, and then we're also going to look at issues that can happen in both of these processes. Okay, so differences that we see in meiosis, we get crossing over. As those homologous chromosomes come near each other, we can get the trading of alleles. We also see that our sister chromatids, so those identical chromosomes, stay together throughout all of meiosis 1, where in mitosis, these separate right away in anaphase. And we see that DNA replication is suppressed between meiosis 1 and meiosis 2. Whereas anytime you get a round of mitosis, you're going to have DNA replication happen beforehand. Okay. And so here we see this crossing over occurring in meiosis and then separating our homologous chromosomes. In mitosis and metaphase, they line up. They're single files, so there's no crossing over. In anaphase, we separate those sister chromatids. Okay. And so here we see these processes. We've got our meiosis 1 and our mitosis, and you can kind of see the difference here, right? We've got our homologous chromosomes lining up in meiosis, not happening in mitosis, single file in metaphase of mitosis. But in meiosis metaphase 1, our homologous chromosomes are next to each other. Okay, and this continues through, and we end up with these 4 unique daughter cells in meiosis, whereas at the end of mitosis, we have these 2 daughter cells that are exactly the same. Okay, so here are some things that you want to make sure that you know. Why do we do meiosis? So we can create sex cells for sexual reproduction. Okay, you want to know each of the phases of meiosis more than just what meiosis 1 and meiosis 2 is here as listed here. You want to know prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. You want to know all of those steps and the things that are happening in each of those steps. Okay, and then at the end of meiosis, we have 4 cells, all haploid, genetically unique, different from one another. Okay. Male and female gametes, eggs and sperm, are different from one another, and they're not formed in the exact same way. Okay. And so we see a sperm here trying to get into the egg cell also shown. And you can see right away the size of these cells is vastly different. Okay, so here's spermatogenesis. And in general, this is meiosis exactly as we've been talking about it. Okay, we do a couple of mitotic divisions from these special spermatogonial stem cells to produce a bunch of cells that can then go through meiosis. And so this primary spermatocyte is going to go through meiosis 1, right? Now we've got our secondary spermatocytes where each has those sister chromatids in it. We separated our homologous chromosomes in meiosis 1. And then we go through meiosis 2 and we end up with 4 genetically unique cells here, all the same size in spermatogenesis. And then some differentiation happens here to give these cells their flagella. Okay, and then the acrosome at the end. But you don't need to worry about that. Basically, spermatogenesis follows the normal meiosis, and we end up with 4 viable sex cells, 4 sperm that can go on and potentially fertilize. Oogenesis is a little bit different. Okay, and through this process, we've got our germinal cell. So that germ cell is going to go through mitosis, create some primary oocytes, and then we go through meiosis. But the thing that we see here is that we no longer get even cytokinesis. One of the cells is going to be much larger than the other. Okay, so one of the cells gets a lot more of that cytoplasm than the other. Then meiosis 2, this first polar body is going to divide, not very important. Second polar body then divides, but again, or the secondary oocyte divides. We create a second polar body and then the ovum. And again, right here, we see that we're not getting equal division through cytokinesis. So at the end of oogenesis, we go from one cell. We still have 4 haploid genetically unique cells, but only one of them is viable as a sex cell. Those 3 polar bodies will not be getting fertilized. Only the ovum has the ability to do that. So that's our big difference between making sperm and eggs. When we go through meiosis 1 and meiosis 2, we're only making 1 good egg cell. Through spermatogenesis, we're making 4 good sperm cells. Okay. You don't really need to worry about this, but lots of sperm cells are made every day. Not nearly as many egg cells are produced by women. Okay. They're a lot more precious, those egg cells. They're worth a whole lot more because they're harder to make. They're much larger. Okay. Fertilization, right? We're not going to talk a lot about this, but the sperm comes into the egg. It makes its way through using various enzymes. That head of the sperm breaks off, and eventually it's going to fuse with that secondary oocyte, and we get our fertilization event where we had 2 haploid cells. Now they come together, and we've got our diploid zygote. Okay. And so we have that single cell zygote, and then the rest of making all of the cells that are you is mitosis. So we do meiosis to make those sex cells. They create a diploid cell, and then that replicates and replicates and replicates, making something like 1 trillion cells that make up you. Okay. Now, this doesn't always go as planned, but we'll go ahead and we'll make another video here to talk about the issues with mitosis and meiosis that can happen.